So today I'm going to tell you my efforts using a unique model system to study how specific cell behaviors are controlled by the gene network. So as a developmental biologist, I'm mostly interested in how the embryos develop, specifically how a complex structure is built upon from a single cell fertilized egg. So to think about that question, so the cells in a developing embryo not only need to be specified with particular cell phase, but they also need to move extensively in the embryo to reach their final destination. To help you appreciate the complexity of cell movements during embryogenesis, I will use a zebrafish gastrula stage embryo as the example. So you can see in a developing zebrafish gastrula, you see different cell populations at different places in embryo and go dramatic different patterns of movements in processes like involution, ingression, epiboly, etc. So how do these cells have these different behaviors? If we look more carefully at the single cell level, we can see that there are several ways to achieve specific modes of cell movements. The cells can undergo movements either as a group in processes like intercalation, invagination, evolution, etc. Alternatively, a group cells can move as individuals but coherently in processes like directed migration or ingression. So regardless of how they do that, so first, they have these different behaviors, and the way to generate this behavior is presumably by the gene network operating in these cells. To study these cell-specific behavior control, so traditionally there have been several approaches. One is a cell biology point of approach, where people study cell behaviors mostly in vitro, basically cell lines, and they just take a few candidates, knock down, and see what changes. But there are some limitations for in vitro study because they could behave differently as they do in intact embryos, and also other complications like the difference in their neighbors. So alternatively, developmental genetics study the cell behavior mostly by forward genetic screens, trying to identify genetic mutations that affect individual cell behavior. But more recently, with advances of this molecular imaging techniques and the high-throughput sequencing screen, we see a convergence of these two approaches. Basically now, what I have been doing is to identify a specific cell behavior and then studying the cell type specification in that cell and trying to understand how the gene network acting in those cells will control cell-specific behaviors. So the next slide actually illustrates the approach to this method. So considering a specific cell type, so the cell type fate is specified by the gene regular network, basically by hierarchy of transcriptional factors. So on the other side, you have a cell biology network. Basically, these are protein-protein interaction networks that will produce specific cell behaviors. But when cell biologists study these questions, they just assume the cell network is there, then every gene is there to do their work. But we know that's not the case, because if every cell type has all the cell biology genes, then how are they different, and why do they have different cell behavior? So another view is that maybe every cell biology gene is controlled by the transcriptional network so that you can modulate the level of each cell biology gene. But is biology really this complicated? So my advisor, Mike Levine, likes to say that the correct biology answer is usually elegant and a simple one. So work from our lab and a few others recently show that the real case is more like this. So within a cell biology gene network, only a subset of genes actually are controlled by a transcriptional network. And these are rate-limiting factors for specific cell behavior. You can manipulate these individual nodes in a large protein network and produce very different cell behaviors. Thus, it's important not to understand the individual functions of these nodes, but also trying to elucidate the interface between the gene network and the cell biology network as this interface. If you can understand how these genes are deployed in different cell types by the transcriptional factors, then you can understand how different cell types and behaviors are generated. So in the next 45 minutes, so I will tell you about 2 parts of my research. In the first part, I will tell you my study on specific cell behavior, the convergent extension of the Ciona Notochord. So first, I will tell you about a whole genome approach trying to identify the transcriptional of the converging Notochord and identification of candidates for the process. In the second part, I will focus on specific signaling pathway, the FGF signaling, how it establishes the cell polarity in the process. And finally, I will discuss how the FGF signaling pathway acting together with a non-canonical Wnt signaling pathway to control convergent extension through an actin dynamics regulator called cofilin. In the second part, I will tell you about my recent studies using high-throughput sequencing technologies for small RNAs in Ciona. So basically what I did is I systematically identified microRNAs from Ciona, and then so incidentally identified a new class of small RNAs. I will talk about the implications of these studies later in my talk. So first, I will introduce a species I work on. So it's called Ciona intestinalis. It's a solitary ascidian species belonging to the Urochordate subphyla. This is what adult Ciona looks like. It's a sessile filter feeder living in the temperate coastal regions around the world, including the San Francisco Bay Area. So if you remember from your undergrad textbooks, they tell you that Urochordates are the most basal chordate groups. However, this view has been changed recently by whole genome molecular phylogeny studies. People now realize that instead, Urochordates actually more closely relate to us. They are sister group to the vertebrates. This places a cephalochordates or the amphioxus to the base of the chordate lineage. This has great implications because the adults, because as you'll see later in my talk, the developmental mechanisms is often quite conserved between Urochordates and vertebrates. So although the adult Ciona does not look like a chordate at all, so the similarities actually is profound at the embryonic stage. So this is a ascidian tadpole stage larvae. And compared to a vertebrate larvae, it's a zebrafish at a similar stage. You see they are all tadpole looking very similar body plans. If you look at the dorsal ventral patterning of the trunk, you see a most dorsally localized nerve cord and the notochord and other tissues. So basically, this conservation of the body plan suggests that Ciona, doesn't suggest, but it basically reflects the fact that ascidians are indeed very closely related to vertebrates. So one of the there are several reasons to study Ciona because one of them is its unique mode of development. So the adults are not adults. So at the end embryogenesis, the Ciona tadpole consists of about 2,500 cells. So what's unique about it is mosaic mode of development. So if you are familiar with this suggests that embryonic cell fates are specified very early on, and then the cell lineages are invariant. So at 110 cell stage, so these different colored blast smears fates are already specified. They will develop along defined lines. So at the end embryogenesis, you know exactly what cells they will give rise to and where these cells will be located within developing embryo. This greatly facilitates lineage tracing and live imaging of these individual cells in the embryo. So there are several advantages of the Ciona system. So first, it has a very simplified genome. So the genome has been sequenced in 2002. The euchromatin region is only 120 megabase. And because it has not undergone the 2 rounds of whole genome duplication as in vertebrates, so it has a very reduced regulatory gene net set and also very compact cis-regulatory regions like the C. elegans genome does. And the second, we have a very powerful technique in the system. It's using electroporation to create transient transgenics. So the way we do this is we mix plasmid DNA with a fertilized embryo and then zap subject them to electroshock, just like you transform a bacteria. Simply by doing this, we can get, in good times, we can get almost 100% transformation, no mosaicism, and perfect development. And we can transform up to like 5 constructs in one experiment. Simply, this technique allows us to do like test 10s or 20s of constructs in just 1 hour. And also, it takes us 24 hours for the embryo to develop through larvae stage. There also exists extensive genomic tools, including sequence genome EST collection in situ databases. And in addition, this already talked about the conservation of the developmental mechanism. This will become clear in the later of my talk. And finally, its lineage-specific development allows us actually to isolate individual cells, study their behavior, as well as for whole genome studies. So with all that, so I will introduce you to the morphogenetic process I study. So this is called the convergent extension. So what exactly is convergent extension? So this describes the process where you have a field of cells. It narrows along one axis while simultaneously elongated along the other perpendicular axis. So then the result is basically turning a broad field into a single row or a long row of cells. So this is a major driving force for the axial formation in chordate embryos. So this is a time-lapse movie showing the development of a Ciona embryo. So you can see as the embryos go through gastrulation and move to the neural stage, the posterior axis start to extend very dramatically, and you have a very long tail forming. And this process of force is mainly generated by the convergent extension of the Notochord. Oh, the larvae will just finally swim away. So at the cellular level, so how do you get this convergent extension? So there are several ways of doing this. For example, you can have direct cell migration toward a midline, or alternatively, the cellular Notochord forms through a medial lateral intercalation. So in this process, the cells become polarized at the medial lateral ends. They form this polarized protrusions, and they provide a driving force. These cells just crawl against each other and zip at the midline to form a single cell row. So people have studied the molecular basis of convergent extension for quite some time. So, okay, what they found is that convergent extension is closely related to another molecular process called planar cell polarity. So what planar cell polarity is you have an epithelial sheet of cells, and these cells all exhibit uniform polarity. For example, in the Drosophila, a wing bristle or the mouth fur coat, all the furs or the hairs point to the same direction. What people found is that genetic mutations affecting convergent extension in mutant zebrafish, mutant embryo like this, will almost always generate the planar cell polarity defect, basically a randomization of this polarity. This suggests that the molecules, pathways that control this process are highly conserved. And what are these molecular steps? So people have known that it's mainly what most people mostly know about the genes actually belong to the non-canonical Wnt pathway. So this is a schematic showing the basic components of non-PCP, the non-canonical Wnt PCP pathway. So you have the non-canonical Wnt ligands like Wnt5 and 11 signals through the receptor and converge on the disheveled protein. So disheveled protein then interacts with several protein molecules. People refer them to as PCP proteins, like the prickle, strabismus, etc. So what these PCP proteins do, and then somehow affect ectocytoskeleton and produce this polarized cell behavior or a polarized structure. So how do these proteins actually produce a polarity? So in the fly wing epithelia, people know that these proteins are asymmetrically localized. So some proteins like the flamingo streamus at the proximal end, while other proteins at the distal ends, they are mutually exclusive. And this molecular symmetry corresponds to the cell symmetry, basically the direction of the bristles. However, in the vertebrate convergent extension process, mainly in the Notochord. So the same set of genes control the process, but people can never observe a molecular asymmetry in these proteins. They are uniformly localized at the membrane. So that raises an important question. So where does this asymmetry of cell behavior, asymmetry in cell behavior come from at the molecular level? If you think about this question, actually, you can break this process into several steps. For example, very first step, you imagine you need some kind of extracellular signal, a positional cue to generate initial polarity. And then you need to transduce this signal into the cell, and this will influence the actin cytoskeleton to produce polarized cell behavior. And finally, and also very important step, is you need to modulate the cell-cell interactions to allow tissue rearrangement to occur. So now I will introduce you to the real process in the Ciona. So these are Ciona progressive older stage Ciona embryos. Can you see the colors? So these are embryos actually electrorated by a Notochord specific enhancer driving GIP. So this is a Notochord plate at the early stage. You see it's pretty broad, and slowly they will undergo medial-lateral intercalation until they form a single cell row. So this process is not cell-autonomous. If I take only the Notochord precursor from the embryo and put them in a dish, they will not form a single cell line. However, if I co-isolate these Notochord precursors with a neuroectoderm in embryonic manipulation like this, so now we can get a more or less normal-looking single row of Notochord cells. You can see the Notochord extend dramatically, and you can account almost 20 cells in a single cell row. So this suggests that indeed this process needs signals from a neighboring tissue to provide either instructional cue or provide the basis for that. But in order to identify these things, so I decided to take a whole genome approach, basically looking for all the genes expressed in a tissue and trying to identify the candidates that can affect this process. So the way we did this is through effect sorting, fluorescence activate cell sorting process. So how I did this? So this is a Ciona embryo transformed with two constructs. The Notochord specific GIP reporter you saw in the previous slide, but another counter selection marker, a mesenchyme driver driving RFP. The reason for having this negative selection because the Notochord reporter is a little leaky in the mesoderm tissues. So we transform these embryos, dissociate into single cells, pass through a FACS machine, and these green-only cells are pure Notochord cells. You can see the purity of the cells by qRT-PCR analysis. So this isolation will give you a wild-type Notochord. You can compare it to the whole genome. But that doesn't tell you actually which genes are required for Notochord fate. So to answer that question, so I added another genetic condition, basically to knock down the Notochord fate and then look for genes that are downregulated. So the way I did this is by using a transgene. So brachyury, the reporter I used in this study is a most upstream transcriptional factor for Notochord specification in Notochord. So I fuse the brachyuridine binding domain to a considered active transcriptional repressor domain, WRPW. And by expressing this brachyury, a dominant-active brachyury in the Notochord, you can see it knocks down the Notochord fate, and also you lose the convergent extension of the Notochord. So by having this genetic condition, actually, we can look for genes that are downregulated when the Notochord phase is lost. So I perform microarray in two developmental stages, the late neural stage, when the cells are undergoing active convergent extension, and also at the late tailbud stage, where the process has almost ended. So we have these three category genes. So these are Notochord-enriched genes, meaning they are upregulating in a wild-type Notochord cell compared to whole embryo. And I have this brachyury target genes, meaning those genes are downregulated in the brachyury dominant-active condition. So when you combine these two group genes together in the Venn diagram, you see they share about half of the genes. So these genes are both upregulated in Notochord and also downregulated when brachyury is knocked down. Those genes are mostly likely associated with Notochord fate as well as behavior. So what are those genes specifically? So I just quickly summarize them in different gene cell processes like actin dynamics, cell adhesion, signaling pathways, and transcriptional factors. And these red genes basically belong to this category. You can see, so first, there are only a very small subset of all the genes expressed in Notochord, about 10% on average in these different categories. So this actually is similar or consistent with the diagram introduced before, basically seeing only a small subset of cell biology genes, other genes actually subject to transcriptional regulation, while most other genes are actually constitutive. So what are these genes in, what are specific are these genes? So for the transcriptional factor, I identify about 24 transcriptional factors, including known ones like the brachyury. So in vertebrate Notochord development, actually people know very few transcriptional factors that's specific for Notochord development. So only know like brachyury and the knot or floating head at the very upstream of the gene cascade. So many of the genes probably play roles in the specification of Notochord later during development. And so using in situ, I show that indeed these genes are sequentially turned on in the developing Notochord, and they probably form a hierarchy and control the downstream targets of the Notochord cell behavior. And for some of the genes you see, they actually express in multiple tissues, like this ZF zinc finger protein, so both in the Notochord and in the nervous system. If you simply do a simple comparison of the Notochord to the whole embryo, the signal will be much lower if we had not had a brachyury knockdown condition. But anyhow, so these are quick candidates to test in Notochord development. But I'm most interested in the cell behavior of the convergent extension process. So these are the genes actually belonging to the cell behavior pathway, like actin dynamics, cell adhesion, and signaling molecules. So to help you better understand these genes, so I plot them this way in different known cell biology processes like actin dynamics, rho gap, Wnt PCP pathway integrin adhesions. So the red genes are upregulated or positive regulated, basically those genes that are shared by the two microRNA conditions. So as you can see, so within each cell biology process, so only individual ones are positively regulated. So you have some good candidates like adhesion molecules, but in the next few slides, I will focus on one specific signaling pathway, the FGF receptor. So Ciona has only one FGF receptor as opposed to 4 in vertebrates. That makes our life a little easier. So the single FGF receptor is strongly expressed in the developing Notochord at the neural stage. So although it seems quite specific for Notochord, but if I look at the MAP kinase pathway downstream of FGF, it's actually not required for the Notochord formation. So I can inhibit the MAP kinase signaling by using this small molecule inhibitor, U126. So you see it abolishes all the DP staining, but the Notochord just forms normally. So this tells us it's not the MAP kinase pathway, but does FGFR play any role in the process? So the answer is yes. So I knew that because when I misexpress a dominant negative form of FGF receptor, it's fused to a venous to visualize the cells, I do get a convergent extension defect. So you see, at the end of embryogenesis, instead of a single row of Notochord cells, you have several rows of cells, and also the cells look weird at higher magnification. So this embryo actually is a mosaic embryo. So only half of the embryo receive the transgene, so visualized by the venous fusion. So these cells, you see, they are round, and they don't seem to undergo the medial lateral intercalation. There are several rows of them. Instead, on the wild type side, you see the cells undergo convergent extension normally and form a single cell row. So this suggests that the cells probably, when they at FGF signaling step, they probably excel autonomously in the process. So this is the overall defects generated by inhibiting FGF activity. But what are the actual exact cellular defects that's contributing to this phenotype? So we can answer that question by doing more cell biology studies. So this is a time-lapse movie of a wild-type Notochord plates. So this is basically a Notochord plate cut open from the embryo and observed with under time-lapse microscopy. So you can, one of the most prominent feature of, you see these cells are constantly sending out these protrusions, these small bulbs out their periphery. So these are actin-based dynamic protrusions, and they provide the driving force for the cell movements. So as the embryo develops, you can see starting from around the Notochord plate, it slowly starts to elongate along this axis. So it's not perfect, so you won't get single cell row, but you get the idea what a normal, a wild-type Notochord plate does. So when the dominant-active FGF receptor is introduced in the cells, you see now these active protrusions are gone. So the cells are still moving actively, but you never see these active protrusions around the periphery. So we think that the loss of these protrusions are basically one of the main reasons that convergent extension is defective. So we can actually quantify this cellular phenotype by looking at single cells. So the way I did this is basically dissociate these embryos into single cells and observe this behavior in a dish. You see the wild type, I don't know if you can see clearly, it forms these transparent protrusions in a media, so just like they do in the wild type intact Notochord plates. So if I put recombinant FGF protein in the media, so now see what the cells do. So they form this huge protrusion, much bigger than they would do in wild type. And now they form at a much higher frequency and all around the cell, suggesting that FGF actually is sufficient to induce the protrusions. And similarly, this effect of FGF protein can be inhibited by co-expression of the dominant-active FGF receptor in these cells. And this is a statistic of the protrusions. So basically, this establishes a role of that FGF is sufficient to induce these protrusions. And so since we're observing these cells in a single dish, so there's no obvious polarity for the cell. But in the intact embryo, actually, these protrusions are not random. They are oriented. So this is a 3D reconstruction of the confocal reconstruction of the Notochord plate stained with fluorine, highlighting the actin structure. So it's kind of difficult to see, but you see these thick actin stainings at the cell boundaries. So if I plot these protrusions or actin stainings along the medial lateral and anterior posterior axis and count their orientation and the number of the protrusions, you see a trend. So in wild-type embryos, you have an average of 0.5 these protrusions per cell boundary. And they are twice likely to orient within 45 degrees of the medial lateral axis versus the anterior posterior axis, suggesting this protrusion actually oriented and polarized. So if dominant-active FGF receptor is put in the Notochord, then you lose both the number of protrusions as well as the polarity. So now they are randomized. So this basically suggests that in wild-type embryo, you have polarized protrusions, which is somehow controlled by the FGF signaling, and this will contribute to the polarized cell behavior. So that raises a question. So since this is a FGF receptor, so where does this polarity come from? So I hypothesize that maybe there's extracellular signal, a positional information, mostly likely by FGF ligand that can contribute to this polarity. So to answer that question, so let's look at the expression pattern of the FGF ligand in Ciona. So again, Ciona has only 6 FGF ligands as opposed to 24 invertebrates. So they have non-overlapping expression patterns, and then quickly found that there's only one FGF ligand, FGF3, that's expressed in the immediate proximity of the Notochord. This is expressed at the most ventral row of the neural tube. I refer to it as the floor plate of the neural tube. So as you can see, it's in situ of FGF, a single line of cells. It's a reporter driving like Z. You have a similar expression. At the gastroneural stage, actually the 2 rows of floor plates have not intercalated. So you see 2 rows of cells. So anyhow, this is the transfer section of the trunk region. You see basic get idea. The FGF is basically sitting at the dorsal midline of the developing Notochord plate, and it could potentially provide a positional information for the Notochord morphogenesis. And indeed, that's the case because the prediction will be if we inhibit the FGF3 function in the neighboring tissue, then we should also get convergent extension defect. This is exactly what I saw. So I inhibit FGF3 function using antisense morpholinos. So this is our preferred way of inhibiting gene function in the system so far. Also, we're trying to develop RNA-based gene knockdown techniques. But anyhow, so these are morpholinos against splice junctions. So we can quantify the level of inhibition by qRT-PCR. And then you see in FGF3 morpholino morphant, so you see a varying degree of convergent extension defects. You see this brachyury GIP labeled Notochord. So in the most mild phenotypes, you see occasionally you see 2 cells in that one cell along the Notochord row. But more severely, you see 2 rows or even 3 rows of cells, suggesting that indeed you inhibit FGF3 function in neighboring tissue, you get a non-cell autonomous defect in the neighboring Notochord. So this basically suggests that there's somehow a gradient, an endogenous gradient. So then the prediction is that, so if we manipulate or perturb that gradient, can we also get a convergent extension defect? So how I did that? So we have a great collection of tissue-specific enhancers. So what I did is I basically drive FGF expression in all the neighboring tissues of a Notochord, including a Notochord driver, a nerve cord, the muscle endoderm. So this is basically the relative position of these different tissues. So in all these cases, you can see that the Notochord convergent extension is somehow defective. You see cells that did not go into the integrate into a single row of cells, but end up wandering in other places. So this suggests that you not only need endogenous FGF3 gradient, but also the gradient needs to be precisely regulated. So this is a model that we have so far based on the results I discussed. So in wild type, you have a dorsal midline source of FGF3 gradient. And this gradient diffuses and into a medial lateral positional information, and that produces polarized actin protrusions. So if you inhibit this gradient, then you get loss of these protrusions. But that's, so far, it's good. It basically tells us where the initial polarity come from. But so the immediate follow-up question is, so how do this positional information get translated into the asymmetric cell behavior? So to answer that question, we need to look at the pathways more carefully. So far, I've told you about how FGF signaling pathway controls a process. So there are several downstream pathways, including the MAP kinase, which we already excluded in the process, but there are a couple other pathways, the PLC pathway and the PI3-AKT pathway. So, but this doesn't clearly tell us how it actually affects actin dynamics. On the right, this is what people know about the non-canonical Wnt pathway and how that controls the cell behavior. So the current idea is that people think it somehow acts through the bunch of small rho GTPases, which are known to be positive actin regulators. So basically, in the next few slides, I'm trying to tell you my recent results, preliminary studies showing how these two pathways together control actin behavior. So one of the first angles I took was actually to look for what actually will produce actin protrusion. So I told you the small GTPs are well-known positive actin dynamic regulators, and people know actually if you put specific small rho GTPs, it will induce protrusion in a cell culture assay. But in the Notochord, actually, we have a very tissue-specific rho called RhoDF. So it's expressed exclusively in the developing Notochord and another very dynamic tissue in the embryo. It's a migrating heart cells. So these heart cells start at the trunk and tail region, actually migrate all the way to the anterior of the head. So these two tissues are most dynamic in the tissue, and they also correlate with RhoDF expression. So previous postdoc, Christian Leonel, when he inhibited RhoDF by morpholino, he saw both the defect in the convergent extension in Notochord as well as in migrating heart cells. And conversely, if we misexpress the considered active form of RhoDF in the heart or in the Notochord, we also get defects, suggesting levels of RhoDF is also required for the cell behavior. But anyhow, so looking at the Notochord cell behavior, So if I put a dominant-active RhoDF or inhibiting RhoDF function in the Notochord cells, you see that's how the cells behave. So now you lose this actin protrusions around the periphery. Basically, it's consistent with how RhoDF works as people get from other systems. And alternatively, if I misexpress a constitutive active form of RhoDF, it produces bigger protrusions at a much higher frequency. It's all consistent, suggesting that the upregulation of RhoDF is indeed correlates with the actin protrusion behavior in the Notochord. But then the question is, maybe is it RhoDF that's being regulated by either these pathways, FGF, or RhoDF, or non-canonical Wnt PCP. So this next few slides are mostly molecular epistasis tests. So if you're not familiar with the idea, it's basically we're trying to put different components of two different pathways and see which one is more dominant against other and trying to establish a cascade of gene activity. So this is what wild-type Notochord cells do in the culture. You have seen a couple of times. And this is a co-expression, a transfection with a dominant-active FGFR receptor. You lose these actin protrusions. So if RhoDF functions downstream of FGF pathway, then prediction is that if I put a constitutive active form of RhoDF back into the cells where the dominant-active FGF receptor is expressed, you could rescue the loss of protrusion phenotype. But do we really see that? So this is how the cells behave. So it's kind of weird looking. So it has this huge protrusion, but it's stable. So you don't get active protrusion like it grows and then it tracks and you form a new protrusion at another place. It does not rescue the protrusions. And similar, RhoDF is transient upregulated. I test another Rho GTP, it's called the RAC1. It's also an important regulator for this process, and it doesn't rescue the phenotype either. So this tells us basically the RhoDF is probably not regulated by the FGF pathway, and the loss of the protrusive phenotype cannot explain by this RhoDF regulation. So then what are the other candidates? So to answer that question, we actually need to look at the regulation actin dynamics more carefully. So this is summary of what people know about the control of actin filament, actin dynamics. So actin filaments are controlled actually by both positive regulators and the negative regulators. At one end, so the new subunits, actin subunits are added, basically actin filament grows and make the protrusions. And the small GTPase, activated small GTPase, Rho GTP is actually required to add this active actin to recruit the new actin subunits. However, the actin dynamics is also controlled by the negative factors. So these are molecules, mostly one of the best known other effector is ADF cofilin. So what cofilin does, it severs the actin filaments from the other end. So it depolarizes the actin and makes new actin subunits. So basically what it does, it's basically disassembles existing filaments and also provide new subunits for actin filament growth in other places of the cell. So actin ADF cofilin molecules basically are a group of small actin depolymerization factors. They are like 170 amino acids in size, and people know that they are mostly regulated by serine 3 phosphorylation. So actin cofilin actively regulate actin dynamics in many systems, and for example, in cancer metastasis, people know that cofilin is upregulated. But anyhow, so what's the surprising finding I got is that if I co-express a constitutive active cofilin, now it can rescue the loss of cell protrusive activity in a dominant-active FGF cell. So you see, compared to the loss of protrusive activities, now you have these protrusions back. Bingo. So this suggests that FGF probably regulates these actin protrusions somehow through the cofilin step. So now we have the FGF, try to have this previous result linking FGF to the cofilin. But what about the non-canonical Wnt pathway? So because that's, before going to that, I'll show you how the wild-type covalent affects the Notochord cell behavior as expected. If you put wild-type covalent in the cell, you get many more protrusions than a wild-type. And conversely, a dominant-active form of covalent will suppress this protrusive activity. And the considered active covalent is basically like what wild-type covalent does. So this is statistics showing the expected behavior of the protrusive activity corresponding to the covalent activity. But anyhow, so the non-canonical Wnt pathway, traditional people think that non-canonical Wnt PCP pathway basically a positive regulator of actin protrusive activity and cell behavior. Basically, it promotes actin dynamics. But what I found is contrary to that. So if I overexpress a wild-type disheveled in Notochord cells, you actually see a loss of this protrusive activity. Instead, when the disheveled activity is inhibited by a dominant-active form of disheveled, now you see a huge increase in the number of these protrusions around the cell. So what's good about it is that this effect seems to be ubiquitous for all the components for the Wnt PCP pathway. So the PKC delta is another member of the Wnt PCP pathway. If I inhibit the PKC delta using a similar dominant-active, you also get this increase of a protrusive activity. And this is a summary of the statistics. So basically, this tells us that somehow the Wnt PCP pathway is negatively regulating the actin protrusive activity. Is that also through covalent or upstream of covalent? So the answer is yes. So you can see that in this molecular epistasis experiment, so loss of protrusive activity by overexpression of wild-type disheveled can be rescued by co-expression of a considered active form of covalent. You get these protrusions back. Converse experiments, the increase of the protrusive activity in dominant-active disheveled conditions can be suppressed by the dominant-active covalent. So these two reciprocal experiments basically establish that disheveled also functions upstream of covalent, but possibly through a negative way. So this is a summary of our model. Basically, what we think now is that covalent is regulated by both of these processes in the Notochord. FGF receptor pathway actively control covalent activity, while the Wnt PCP pathway actually negatively regulate covalent activity. And remember, in the Notochord, covalent itself expression is not regulated, so it's constitutive. However, so you have the positive regulator of actin dynamics, the RhoDF, that's transient upregulated. Together, you have both active and negative regulator of the process. Together, they will form this active or dynamic actin dynamics in the Notochord cells. And the very last part is that RhoDF, as well as other individual components of both FGF receptor pathway and the PCP pathway, are subject to transcriptional regulation by genes like BRCA and a few other downstream transcriptional factors. So this suggests that basically how you beautifully you see the interplay between the transcriptional network and also a cell biology network, the protein-protein interaction networks. Together, they can control a polarized cell behavior in the process. So this is a summary of the Notochord projects I have talked about so far. So first, I use microRNA to identify over 100 candidate genes for both the Notochord specification and cell behavior. They're candidates for gene function studies. And I also show that how the FGF signaling pathway established the initial polarity in the convergent extension process. And finally, I showed some preliminary results showing that how the two pathways together acting through the covalent to control actin dynamics within Notochord cells. So now I will switch gears to talk about the small RNA studies in the next 10 minutes or so. So one of the reasons that promotes me to do the projects is to find the new regulators of cell behavior control. So in the past 10 years, people have realized that the microRNAs are important regulators of many developmental and disease processes. And we figure that maybe you have additional components from the microRNAs and can affect cell-specific behaviors. And also the second goal was actually to develop tissue-specific RNA interference to knock down genes in the Ciona. And to do that, we actually need to understand more how small RNAs actually biology work in the system. So this is the overview of the microRNA biogenesis if you are not very familiar with the field. So microRNA is actually produced by RNA polymerase II from long transcripts as all the other protein coding genes. So these precursors called the prime microRNAs are processed by the Joshua complex to produce a stem loop hairpin of pre-microRNA. So this stem loop hairpin is transported into cytoplasm, processed by the dicer to produce the microRNA and microstar duplex. This enters the RISC complex and suppress gene function. So how do the microRNAs do that? So there are several ways people actually some people think one is more prominent than others. But anyway, they can either inhibit translation, of course, they bind to the 3' UTR, and sometimes they inhibit the translation, and also in other cases, they will introduce promotes the degradation of the transcript. Either way, the end result is the downregulation of gene activity. So to identify microRNAs is not trivial. So I decided to take a basic experimental approach, basically by cloning the small RNA libraries from the embryos and using Illumina high-throughput sequencing to get all the small RNAs and try to identify the microRNA from the library. But that's a challenging task. So you have millions of reads, but which percentage of actually microRNAs? It's very hard to actually distinguish microRNAs from non-microRNAs as you go through the list. So to address that question, so I work with a bioinformatician in lab, Dave Hendricks, to develop our in-house algorithm called MIR-TRAP to predict microRNAs from my high-throughput sequencing data. I'll not go into the details how the program works, but simply by saying, so it's based by the structure function, basically biochemical processing of the microRNA locus. Basically, we are trying to identify the candidate loci that's the most consistent with microRNA biogenesis. All these algorithms complicated, but in the end, our program predicted about almost 400 candidate microRNAs from hundreds of thousand instances. It has very low false negative rates and acceptable false positive rates. And what's comforting is that our prediction identified over 99% of the previous known microRNAs based on homology. So it's basically quite zero. So just to briefly go over our prediction. So this is a molecular phylogeny of microRNA families. So people have known that the microRNA families actually correspond quite well with the phylogeny of different animal groups. It's consistent with basically evolutionary history of different lineages. So these red dots represent new microRNA families predicted by our program. So you see, so before our study, so many of them are missing. So we don't know whether these microRNAs are specific to the vertebrates or whether they have deeper evolutionary origins. So far, our prediction identified over 19 new microRNA families that are existing in the vertebrates. This basically places the evolutionary origin of this microRNAs deeper. Basically, they could come from after the split between amphioxus and the ascidian and the vertebrates, but also that requires further investigation of amphioxus microRNA genes. But anyhow, so in the next 5 slides, I will talk about somehow incident or serendipitous discovery from our study. So basically, from our high-throughput sequence library, we found a new group of small RNAs. So this is what a typical microRNA look like. You have two distinct products. So these are basically high-throughput sequence reads plot against the genome. So you have one strand, one product on one strand, and one product on the opposite strand. They are referred to as 5' mir or 3' mir. So this is basically what people know about the microRNA. But from our study, we saw that over 50% of the predicted microRNA loci actually have two additional products, these and this. So they immediately flank the microRNA and the microRNA star products. And because they're unique position, we name them microRNA-offset-RNAs. My came is with this name, more. Basically, you have more RNAs. So anyhow, so this is a typical microRNA locus, the miR-133 locus. We see this is the miR, a 5' miR product, 3' miR star. Sometimes you have an intervening loop. But in this particular case, you have this 5' immediate flanking product of 5' more, which is even more abundant than the mature microRNA sequence at late stage of embryogenesis. So this product, if you plot at the stem loop hairpin of the microRNA precursor, you see that it immediately flanks the microRNA and microstar duplex. So part of it is paired or part of falls in a loop sequence. So using northern analysis, I show that indeed these products are real and distinct bands in the northern plots as what we saw in the high super sequencing library. So this is northern against the mature miR-133 and the MOR-133. You can, in different embryonic stages, you see accumulation of both the miR-133 and the MOR-miR-133 products. And what's interesting is that these products seem to be co-produced. So I say that because if I overexpress this locus in the embryo, so I see both the increase of the mature microRNA sequence as well as the MOR-133 sequence, so that they are probably produced in a more or less in the same biochemical step. So because Ciona, so these products are so abundant in Ciona, so we reason that maybe you could see that in other species as well. So during our study, so another postdoc in the lab who studies Drosophila small RNAs, he was sequencing several libraries. And in this one particular microRNA loci consisting of 8 microRNAs, he also saw this, clearly he saw this flanking product, more products. But they are very rare. For example, you see here you just see like 30 copies out of over 1 million reads. And because they are so rare in the Ciona, he first saw that's just junk from the sequencing process. But after we saw the highly abundant product in the Ciona, we think it's probably real. But why is it so abundant in Ciona while not in other species? So one hypothesis is maybe the Ciona microRNA processing machinery is just unique. So for whatever reason, it just produces these more products. So is that really the case? If that's the prediction, then if we take the Drosophila microRNA locus and put in the Ciona, then the expectation, you certainly get more these flanking products. This is the actual case. So this is a small library made from a Ciona transfected with this fly microRNA loci. So now you see a sudden increase of this flanking product. For that, instead of 36 copies, you have over 1,000 copies, almost the same abundance as a mature microRNA sequence. And now, since they are overproduced, now we can actually detect them by northern analysis in a graph like this, suggesting that indeed they are real and they are they are meaningful bioprocessing products from the locus. So based on all these knowledge, we have several models basically trying to suggest how you can produce more from a microRNA locus. So traditionally, the canonical model is shown in this. People have this, we can ignore the details, basically how the Joshua and the partial complex cleaves the microRNA at the stem and produce the microRNA star duplex. But in our model, we propose several alternative ways. Maybe there are multiple subunits of the Joshua complex and can do more than one cleavage of the prime microRNA precursor and produce this distinct microRNA and more products. So far, we see these products in both the Ciona and the Drosophila, but after our identification of people start to see them more in other species, including the stem cells, the human brain libraries, and we even saw that in viruses, suggesting it's a very common feature of the process. We think it's probably ubiquitous step from the microRNA processing. Okay, so in the next few slides, I'll talk about future plans since we don't have a chalk talk, so I'll go a little over more detail. So I hope I've convinced you so far that Ciona is a very open system. There are many open questions, and it's easy to pursue these individual values, even from the research I just discussed. I have quite broad interests. Some of them are immediate extension of my experiment. Some are kind of long-term goals, trying to expand into whole genome studies. So first, I want to basically finish the co-filling and the regulation activity project, basically showing how the FGF and Wnt PCP pathway can control co-filling at a biochemical level. I'm trying to address this question using the xenopus as a model system to substantiate the findings in Ciona. And second, I told you I have hundreds of candidates for Notochord development in Ciona from the microRNA study, and we can do quickly reverse genetics and candidate approach for gene function. And more interestingly, actually, I want to use a whole genome approach actually to ask the evolutionary origin of the Notochord. So I will discuss that in the next slide. And finally, I want to study the transcriptional landscape of the different cell lineages. Basically, so the cells have the same genome, but they have very different developmental programs that's manifested by different gene networks that unfold in each cell type. So with this, because we can do lineage-specific studies, we can actually ask that question easily. And finally, follow-up for the microRNA study, I want to study the tissue-specific expression and function of these microRNAs. So this is just a summary. This is a repeat of the networks and the candidates for the microRNA for the Notochord microRNA study. Sorry. So I want to show this basically to tell you that actually I have preliminary results suggesting that many of these red genes, transcriptional regulator genes, actually do produce Notochord phenotypes. So for example, this one interesting ephrin receptor. So ephrin is required for cell-cell interactions. And I show that actually inhibiting ephrin activity will result in convergent extension. So this phenotype actually is different from inhibiting FGF receptor because the cellular protrusive activity are normal. However, the cells just seem to lose contact and adhesions, and you get this similar extension, convergent extension defects. And more interesting for genes in the cell adhesion pathway, so actually that's one published data showing they identify, Bill Smith's group at UC Santa Barbara, they identify a naturally occurring mutation in laminin A5 that will result in convergent extension defects. So laminin is extracellular matrix protein, and I have preliminary data suggesting the intracellular adapter molecule, tailin, which actually signal between the integrin receptor and the intracellular actin dynamic network. Inhibiting tailin function will also result in convergent extension defects. So basically show that we have many genes that are required for Notochord expansion identified by microRNA. But now this interesting question about the evolutionary origin of Notochord. So Ciona, as well as vertebrates, still belong to the chordate group. So the name chordate comes from the chord with all the Notochord. So Notochord is a defining feature of the chordate phyla. So it developed from the dorsal-most mesoderm tissue. But in non-chordate species, people have been unable to identify like a similar homologous tissue. So basically, they don't know where does the Notochord evolve, where does it come from. So traditional people do this evo-devo study. They just pick a few candidate genes and show the similar expression and say, that's the tissue is homologous and conserved. But with the development of these whole genome tools technology, now we can actually integrate the tissue at the whole genome level and look for all the gene expression profiles as well as the gene regulatory network in the tissue and trying to understand what are the conserved genes and the conserved regulatory toolkits that specify a similar tissue in different animal groups. So I propose to study the Notochord development evolution in 3 representative chordate species, the Ciona of my favorite, and the zebrafish. And for this worm-like creature, if you don't know what it is, actually it's an Amphioxus, the most basal chordate. So we have technique actually to isolate Notochord cells from both the Ciona and the zebrafish. And for this really non-canonical model system is a challenge. I'm collaborating with a lab in Taiwan actually to develop a tissue-specific labeling and isolation technique to get Notochord cells from amphioxus. And once we have that, we'll do transcriptional analysis and also the ChIP-seq against several of the most upstream transcriptional factors in the tissue. This is just a side note. So this is, if you know what this is, it's called acorn worm or hemichordate. People think it's the most closely related non-chordate species. Former postdoc from Mike's lab, Chris Lowe at Hopkins Marine Station at Stanford studied this. So some people think actually this is a precursor of the chordate larva during evolution. But anyway. So to extend the whole genome study, so I want to do actually a tissue-specific, identify the tissue-specific transcriptional landscape of the Ciona lineages. So there are 7 major tissue types in the Ciona, main tissue types. We can identify and isolate tissues by different tissue-specific enhancers. So these are GIP, enhancer driving GIP. This is epidermis, the central nervous system, the notochord, the migrating heart cells, mesoderm, tail muscle, and the endoderm. So we have these markers which allow us to do FACS sorting, get pure cell populations, and then we can do several of these hydrogen sequencing studies like RNA-seq, POL2 chip, and also the epigenetic markers. Basically try to identify different transcriptional programs in different tissue and trying to understand which tissues or how each tissue is specified by the gene networks, and how similar and different they become during embryonic development. So this final slide actually summarizes my approach to understand this question, the interface or relationship between a gene network and cell behavior. So within a cell type, in the nucleus, you have different transcriptional factors controlling the gene expression. And we can ask those questions details by ChIP-seq against the transcriptional factors, POL2 chip for gene activity, and RNA-seq for the mRNA expression patterns. But outside the nucleus, so in the cytoplasm, you have receptor molecules which receive signal from outside the cell. So it will transduce the signal inside the cell, either directly feeding into the transcriptional program, alternatively or in parallel. It can feed the information into other cytoplasmic components, like a cell biology genes, basically by protein-protein interactions. And we can do that by proteomics or 2 hybrid screening, etc. So these cell-cell protein-protein interactions will ultimately generate specific cell behaviors, and we can do that by different live imaging and all this microscopy technique. And finally, we have this new group of regulators, microRNAs, can feed into any of these processes. So this is basically what I have for today. So I will to acknowledge my collaborators. For the Notochord FGF signaling project, I collaborate with Richard Hart lab, the Xanthus lab, graduate student Sarah Pugh helping with some of frog experiments, and Edwin Monroe at UChicago helping with the live imaging. For the small R analysis, I had the privilege to work with bioinformatician Dave Hendricks and another postdoc LiPo Ben Haley. So for the Notochord microanalysis, a former postdoc, Lionel Christian, who is now at NYU, helping with analysis. And finally, I want to thank my advisor, Mike Levine. So he's a very unique way advisor. So not only he provides scientific guidance, but also various motivations. So by leaving this question mark, asking our whereabouts. So anyway, so this is my bench. Someone sitting there. And Mike left the notes. Okay, thanks.
Okay, we'll now have questions. Are there any questions for Dr. Sheets?
So you mentioned that the experiments are done by transient transgenic.
Mm-hmm.
So that you make no stops in this.
Right. So for transient genetically, we can get expression up to like 4 days. That's like young juveniles. But for stable transgenic, we need to do actually integration using like recombinase and some kind of transposes. Yeah, there are ways to make transgenics. But to maintain the stock, you need have access to a like marine station. There's one at UC Santa Barbara, which is funded by NIH. So they have culture facility. We just keep the transgenic animals there, and they send us a sperm, to do experiment.
So with it, you have to find a
for the reverse, transgenic, Yeah, but it's well lucky because for the molecule we're most interested in transcriptional factors and cell biology genes, you can usually make a dominant-active form quite easily. But if that's not the case, then we need to do morpholinos or something.
I noticed when looking at the phenotype of Notochord development, that there seemed to be a slight difference between FGFR knockouts and fulfilling knockouts. It seemed as if the FGFR ones seemed to still, although they weren't along this nice linear pattern that you expected, but they still seemed to be kind of in somewhat of a pattern, which didn't seem to be the case for the fulfilling. Any idea why that might be the case?
So the co-filling actually phenotype I saw, they were mostly in isolated cells. So I haven't shown you like intact co-filling, dominant co-filling in an intact embryo. Actually, it's probably milder than the FGF phenotype. We think it's because it's true for all these dominant-active phenotypes when doing the cell biology genes. So you have stronger phenotype when looking at individual cells compared to like in a whole embryo. So I think probably has more to do with the penetrance thing, and also because our electroporation method will be subtle differences. One cell probably expresses the higher levels than the neighboring cell. So you can even see that in the reporter, right? Some cells are brighter than others. So we think it's probably a variation by the transgenesis.
So you don't think there's necessarily a difference based on genes? It just depends on penetrance?
I think it could be both, but now we think the FGF probably mainly regulates the cell behavior through co-filling. I'm trying to test that in the frog, because actually some data I didn't show. So I will show that actually inhibiting FGF activity will actually reduce co-filling and localization in the cell.
Are those constructs or mutations that have been identified as
Yes.
Yeah. So there's no question that they're
Correct. Yeah, we try to make the dominant according to published data. Basically, this is just you truncate all the intracellular space.
Do you know is the level of expression of these things comparable to wild type, or is it saturated?
Okay, way more higher than wild type. So it's because I have done some like rough quantification. I think in a cell probably has like 50 transfusionation events compared to a wild type. You only have about two. So it's it's kind of hard to control the exact level, but we can do that. But it's normally that's not what we do. We just saturate the cells.
So what exactly are they? So these have the extracellular?
Right. Actually, they don't integrate. So that's why you lose them along the cell divisions.
I look at your movies and correct me if I'm wrong, but it looked to me like you were seeing not more events, but the protrusions were not recycling quickly. They would last longer. I mean, maybe with the sped up films, it's difficult to see differences in frequency. But it looked to me like the overall activity was similar. The more striking thing was the more extensive size of them before they were recycled.
I think it's a combination of phenotypes of both. Basically, it's both the size of protrusion, also the half-life of protrusion. So that's why I'm doing experiment in frogs, because the frog, the dorsal marginal zone, which is equivalent to the caudal mesoderm so it's bigger and it's easier to image over longer time span and do the quantification. So these are isolated cells on a slide. So they are, because we don't have a good environmentally controlled chamber. So there's more variation in our quantification than I would like to have. Sorry, what's sorry?
In a cell culture, those cells in the case of the wild type, particular state that you have to be
So for the isolate individual cells, so we cannot tell. So basically, I just identify them by the expression of the GIP marker. So I try to actually do them at around the same stage, like the late neural stage, equivalent to an intact embryo. Then I image them maybe within a half-hour span. So they are more or less at that stage, but they do that, as I mentioned, yeah, there's definitely a lot more variation than, yeah, the controlled, setup. But overall, because for this problem, it's just I got this observation in the Ciona, and I want to test them more rigorously in the frog.
Okay, if there are no further questions, let's thank Dr.